class. Hello students, I am Hema Rajesh, tutor for biology. I will be covering biomolecules today. Briefly, I will run through how to access revision notes and test in clay6.com. So go to clay6.com, click on practice. There you will find many packages. Select a package. Today I am selecting NEET Premium since we are doing NEET classes. Now here you will find tests for physics, chemistry, biology and NEET full test. And if you click on biology 11th, you will see chapter wise tests. All the tests are done depending upon its importance in NEET exams and also the complexity. Here you will find that every chapter has got different levels of tests. Now if you look at these bubbles over here, you will find that it has got a green bubble. Green bubble indicates that you have already completed the test, but you can always visit and redo the test once again if you feel you want to take it again. Suppose you have scored less marks. You want to improve your scores after 2 or 3 days, you are most welcome to do it again. The blue bubble indicates that you are yet to take the test. Red bubble is a randomized test. After you complete all the levels, if you feel you want to test where you stand, then you can take up this test. Yellow button shows that you are yet to complete the test. Sometimes you would have started the test. After 3 or 4 questions, you would have felt you are not adequately prepared for the test, and then you would like to end the test. So once you revise and you feel that you have prepared and you want to test your level of understanding, you can get back to this yellow bubble and continue the test. Now I'm sure that you're all getting tensed or gearing up for your NEET exams. Now you might be wondering what are the different kinds of questions that come up. If we look at the NEET pattern, it is very clearly we are able to differentiate three kinds of questions. One is NCERT based question, second is direct from the previous year's question papers, and third modified questions. Now NCERT based approximately 68 to 70 questions in the examination are directly from NCERT books. They can be attempted easily by those who read NCERT syllabus religiously as though it's a holy Bible. Now, direct from the previous year's question papers, another 8 to 10 questions in the examination can be solved if you have solved NEET question papers of previous years. These questions are repeated with little or no modifications. Modified questions, at least 8 to 10 questions in the NEET will be newer questions. These questions become the game changer for those, as these can't be solved by mere mugging up of facts. Though they are from NCERT book, to get them right, you need to apply the knowledge that you have about the subject. So in this website, an earnest attempt has been made to help you prepare adequately for your NEET. I hope you have now got the framework. Now here you find the revision notes. If you click on this, it opens into a PDF like this. And this PDF is just about 135 pages, but includes all the important points that are essential for your NEET exams. And this is an valuable asset for your NEET preparation if you take this package. And if you take tests, that will really help you, and you will also see how this PDF would help you correlate with your preparation and also in your test. So the test will help you to prepare NCERT text without much effort, so that as the questions are created looking into the need of the NEET examination. Now let me begin with the lesson, biomolecules. So all the elements present in the sample of the Earth's crust are also present in the sample of a living tissue. Closer examination has revealed abundance of carbon and hydrogen with respect to other elements. All carbon compounds obtained from living tissues are called biomolecules. Now, biomolecules whose molecular weights are less than 1000 Dalton are referred to as micromolecules or biomicromolecules. Now let us have a look at these biomicromolecules. The first one is the amino acid. Organic compounds containing an amino group and an acidic group as substituent on the same carbon, that is alpha carbon. Hence they are called alpha amino acids. This is the structure of the amino acid. So this is the structure of the amino acid. This is nothing but a substituted methane. There are four substituent groups occupying the four valence position: hydrogen, carboxyl group, amino group, and the R group. This R group will vary for different amino acids. Now this is the important key points. Now there are 20 amino acids which you should learn: lysine, valine, arginine, histidine, glutamic acid, methionine, glycine, proline, isoleucine, glutamine, aspartic acid, cysteine, threonine, histidine, phenylalanine, tyrosine, asparagine, glycine, and serine. Now, from the NEET point, when we have analyzed, this is how it has been classified into amino acids. Acidic amino acids of the 20 are aspartic acid, glutamic acid, asparagine, glutamine. Basic amino acids are arginine, lysine, histidine. Aromatic amino acids are tyrosine, phenylalanine, and tryptophan. Cyclic amino acid is proline. Hydroxyl or sulfur or selenium containing amino acids are serine, cysteine, selenocysteine, threonine, and methionine. Aliphatic amino acids are glycine, alanine, valine, leucine, and isoleucine. So you can make a note of this. I give you 1 minute. Okay. Now with this amino acids, now let us look into the next thing, that is the micromolecule would be lipids. You might wonder why lipids are micromolecules. Yes, because the molecular weight is less than 1000 Daltons, but in the retentate they are found in the acid soluble pool, and that is why they come under the part of organic compounds. Now let us look at lipids. Lipids are generally water insoluble. They could be simple fatty acids. You know, a fatty acid has a carboxyl group attached to an R group. R group could be methyl or ethyl or a higher number of a methyl group. Carbon atoms will range from 1 to 19 carbon. Example, if you take palmitic acid, it has 16 carbons including carboxyl carbon. Fatty acids could be saturated or unsaturated. So if it has got a single bond, then it is a saturated fatty acid. Now, if it is unsaturated, it will have one or more double bonds. Example for saturated is butyric acid, palmitic acid. Unsaturated, it is oleic acid and linolenic acid. Another simple lipid is the glycerol. So glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Fatty acids get esterified with glycerol. They can form monoglycerides, diglycerides, or triglycerides. They are also classified as fats and oils based on their melting points. Oils have lower melting point and remain as liquids. Example, gingerly oil. Fats, on the other hand, can become solidified at lower temperature. Example, coconut oil and ghee. Some lipids have phosphorus and phosphorylated organic compound in them, and they are referred to as phospholipids. Example, lecithin. Now, let us come back to metabolites. So the one next part of this would be the metabolites before we get into the biomacromolecules. So what are the different metabolites you have to understand about? So you know there are two kinds of metabolites. Metabolites are nothing but intermediate products of metabolism. A metabolite is usually restricted to a small molecule. A primary metabolite is directly involved in the normal growth, development, and reproduction. Primary metabolite performs a normal physiological function. Example of primary metabolites are carbohydrates, proteins, and lipids. Secondary metabolites are organic compounds that are not directly involved in normal growth, development, or reproduction. They have important functions. If secondary metabolites are absent, they may harm the organism. So they have important ecological functions. And if they are absent, they harm the organism. Now, important secondary metabolites, you had to make a note of it. Pigments, carotenoids, and anthocyanins, alkaloids, morphine and codeine, terpenoids, monoterpenes and diterpenes, essential oils, lemongrass, toxins, abrin and resin, lectins, concavalin, drugs, vinblastine and curcumin. Actually, vinblastine and curcumin are also alkaloids, polymeric substances, rubber, gum, and cellulose. This will be available in your textbook as a table, but generally we tend to overlook it, but then you will be having questions from that. Now let us look into the average composition in a cell. If you look at water, it is 70 to 90%, proteins 10 to 15%, carbohydrates 3%, lipids 2%, nucleic acid 5 to 7%, ions it is 1%. Okay. Now let us get back to macromolecules. So the macromolecules you will be looking at first is the proteins. Now you know proteins are polypeptides. They are linear chain of amino acids linked by peptide bonds. Each protein is a polymer of amino acids. A protein is a heteropolymer. That is, it is made up of different kinds of monomers. Collagen is the most abundant protein in the animal world. So this question has been asked repeatedly. The most abundant protein in the animal kingdom, it is the collagen. Rubisco is the most abundant protein in the whole of the biosphere. So you have to look at the question properly. The choices will be given, collagen and Rubisco will be included, but you have to see what the question really is asked for. Is it for the animal kingdom alone? Then it is collagen. If it is for irrespective of any kingdom or in the whole of the world, in the whole of the biosphere, then your choice has to be Rubisco. Now let us look at the structure of the proteins. Now proteins can be studied structurally at 4 levels: primary structure, secondary structure, tertiary structure, and quaternary structure. Now primary structure describes the order of amino acids that are joined to make the protein. Now primary, this first amino acid in the primary structure is called N-terminal, and the last is called the C-terminal amino acid. Now in the primary structure is usually we use abbreviations to represent the amino acid residues. So in the primary structure, the linear sequence of amino acids, what are all the amino acids had to come in to form a particular amino acid, will be represented by 3 letters. For example, TRY dash GLY dash PRO dash VAL dash GLY. That means to say it is trypsin, glycine, proline, valine, and glycine. So this is the primary structure. Now example is insulin. It is composed of 51 amino acids in 2 chains. One chain has 31 amino acids and other has got 20 amino acids. Now let us look at secondary structure. Now for the secondary structure, yeah, you can visualize this is the secondary structure and this is the tertiary structure. Within the long chains, there are regions in which chains are organized into regular structures known as alpha helices and beta pleated sheets. These form the secondary structure of the proteins. Secondary structures are held together by hydrogen bonds. In an alpha helix, the protein chain is coiled like a loosely coiled spring. In a beta pleated sheet, the chains are folded so that they lie close to they lie side by side to each other. Now tertiary structure, final specific geometric shape that a protein assumes will be your tertiary structure. This is determined by a variety of bonding interactions between the side chains of the amino acids. These bonding interactions may be stronger than the hydrogen bonds between the amide group holding the helical structure. As a result, bonding interactions between the side chains may cause number of folds, bends, and loops in the protein chain. Different fragments of the same chain may become bonded together. There are four types of interactions between the side chains: hydrogen bonding, salt bridges, disulfide bonds, and non-polar hydrophobic interactions. And finally, it will be the quaternary structure. So in the quaternary structure, two or more polypeptides will fold and arrange with respect to each other. It is the three-dimensional structure of a multi-subunit protein and how these subunits fit together. It involves clustering of several individual peptides or protein chains into final specific shape. A variety of interactions here also is seen by hydrogen bonds, salt bridges, disulfide bonds, as they hold various chains in a particular geometry. Now, there are two major kinds of proteins of quaternary structures, namely fibrous protein and globular protein. Now, fibrous protein, example keratin, silk proteins, globular proteins, example insulin, hemoglobin, and most enzymes. So, this is about the proteins. Now let us look at the nucleic acids. Now you know nucleic acids means immediately what flashes to our mind is DNA and RNA. So it is another macromolecule. DNA stands for deoxyribonucleic acid. RNA stands for ribose nucleic acid. Now this is made up of a nitrogenous base and sugar and a phosphate. Now let us look at the nucleic acid. They are polynucleotides. They make the macromolecular function together with polypeptides and polysaccharides. Now, building block is a nucleotide. A nucleotide has got chemically three distinct components. One is a heterocyclic compound, nitrogenous base. Second is a monosaccharide, a sugar, which could be a ribose sugar in case of RNA or deoxyribose sugar in case of DNA. And third is the phosphoric acid or phosphate group, which could be monophosphate, diphosphate, or triphosphate. If you remember your ATP, it is nothing but a nucleotide, that is adenosine triphosphate. Now, nucleic acid containing ribose sugar is called ribonucleic acid, as we said, and without it, it is called as the deoxyribose nucleic acid. Now, let us look at the nitrogen bases here. The nitrogen bases are divided into two, purine and pyrimidine, depending upon the ring structure. Purine are single ring structure, pyrimidine are double ring. Adenine, guanine, pyrimidine, cytosine, thymine, and uracil. Now, if you look at the nucleoside, nucleoside is made up of nitrogenous base plus sugar, and that nucleoside is called adenosine, guanosine, cytidine, thymosin, and uridine. See, this in this pyrimidine, cytosine, and thymine are found in DNA. Uracil is found in the RNA along with adenine, guanine, and cytosine. Now, let us look at the nucleotide. Now, if in the nucleotide is nothing but nitrogenous base plus sugar plus phosphate, this they can also ask you like nucleoside plus phosphate, because nucleoside is nitrogenous base plus sugar. Nucleotide is nothing but nitrogenous base plus sugar plus phosphate. So it is nothing but nucleoside plus sugar. So adenyllic acid, guanylic acid, cytidyllic acid, thymidyllic acid, and uridyllic acid. So the next polymer we are supposed to know, macromolecule, is the polysaccharides. Polysaccharides are very long chains of sugars. They are threads containing different monosaccharides as the building blocks. Example, cellulose. It's a homopolymer made up of repeated units of glucose. Starch is the reserve food material of the plants, and glycogen is the reserve food materials in the animals. Inulin is a polymer of fructose. Starch forms secondary helical structures and therefore holds the iodine molecule in the helices. Thus, the complex starch iodine appears blue. Cellulose does not contain complex helices and hence cannot hold iodine. Exoskeleton of arthropods have a complex polysaccharide called chitin, a heteropolymer. So if you look at chitin, it is chemically N-acetyl galactose amine, and this forms the exoskeleton of the arthropods. Now, before I forgot, let us look into proteins and their functions. This is very important. I'm sorry I had to digress. Now, collagen, intercellular ground substances are present. Trypsin, it's an enzyme. Insulin is an hormone. Antibody fights against infectious agents. Receptor, sensory reception. GLUT4 enables glucose transport into cells. This is very, very important because they have asked in most of the exams, they'll ask you as GLUT1, 2, 3, or 4 enables glucose transport into cells. It is GLUT4, and you have to remember. Now let us look at the pathways. Now the pathways are anabolic pathways and catabolic pathways. Now anabolic pathway means a building up process. So it is a metabolic pathway that leads to the formation of a complex structure from simpler ones. Now example is acetic acid becomes cholesterol, formation of glycogen from glucose monomer, formation of hemoglobin, carbon fixation in plants to form carbohydrates, and these reactions consume energy. So anabolic pathways, they are endothermic in nature and they absorb the energy, and these are the examples. So the building up pathway of metabolism is known as anabolic pathway. The next one is the catabolic pathway. Metabolic pathways that lead to the formation of a simple structure from a compound structure are called catabolic pathways. Example, glucose is degraded to lactic acid in skeletal muscles, Krebs cycle, cellular respiration, digestion. These are all the catabolic pathways. And here energy is released during the process. So these reactions are exothermic in nature. Now let us look how the bonds are formed linking monomers in a polymer. So one is the peptide. Now this peptide is formed between carbonyl group of one amino acid and the amino group of the next amino acid with the elimination of a water molecule. So it is basically a dehydration reaction where the water molecule is removed. Now glycosidic bond. So you can see this is how the formation of a peptide bond occurs. So from this amino group you will find the removal of hydrogen. From carboxyl group, OH group is removed. So H2O is lost, and this results in the formation of a peptide bond linking two adjacent amino acids. Now this is the glycosidic bond. Now what is glycosidic bond? Bond formed between two carbon atoms of two adjacent monosaccharides, and phosphodiester bond is a bond formed in nucleic acid wherein the phosphate moiety links the 3 carbon sugar of nucleoside to the 5' carbon of the sugar at the succeeding nucleotide. So here you are finding this as the glycosidic bond, and this is a phosphodiester bond. So between a 5' end and a 3' end, you will be finding the formation of a bond. So this is called as a phosphodiester bond. Now with this we have studied about the biomolecules. Now let us get into the enzymes. Now this is very, very important, and it is also very, very crucial that you learn more about enzyme reactions because most of the questions asked are from this part. Now you know what are enzymes are nothing but biocatalysts. They have primary, secondary and tertiary structures because most of the enzymes are proteins, but all proteins are not enzymes. That you have to understand. They have primary, secondary and tertiary structures. Now in the crevice or is a crevice, active site is a crevice or a pocket into which a substrate will fit, just like a lock and key. Now enzymes, you know, when you heat it to higher temperatures, your enzymes get denatured. That is why when you fall sick, your doctor will advise you to eat mild food like idli or bread, because your digestive enzymes would have got denatured slightly because of the higher temperature. At that point of time, doctor will ask you to eat very soft foods that are easily digestible, and you are not allowed to eat high fat rich foods because enzymes would have got denatured. Now, enzymes are isolated that are isolated from thermophilic organs are thermostable. There are certain bacteria, Thermoacidus aquaticus, that can yield you the enzymes, and these enzymes that are obtained from these bacteria are stable even at the higher temperatures. Now, they accelerate the reactions manifold. They lower the activation energy, and enzymes form enzyme substrate complex first and later breaks into enzyme in the product. So this is the preview of how about the enzyme action. Now let me explain to you using the graph of the enzyme action. So how do enzymes bring about such high rates of chemical reaction? The compound that is converted into the product is called as the substrate, that you should understand. Enzymes are, that is, the proteins with all three-dimensional structures, including an active site, converts a substrate into the product, which is symbolically represented as S, and the product is represented as the P. Now, substrate S has to bind to the enzyme at its active site within a given cleft or a pocket. Substrate has to diffuse towards this active site, and the substrate and the enzyme action active site will be like a lock and a key fit arrangement, or it's just like a jigsaw puzzle. It will go and fix it completely. An enzyme substrate complex is formed as an intermediate, and it is a transient phenomenon. When the substrate is bound to the enzyme's active site, a new structure of substrate called transition state structure is formed. Now, expected bond breaking or making is completed, and the product will be released from the active site. Now, structure of the substrate gets transformed into the structure of the product now. Now, this could be more altered structural states between the stable substrate and the product. Now, you look at in this graph, x-axis shows the progress of reaction, y-axis gives you the potential energy. Now, graph represents the progression of the structural transformation of the state to the transition state. Y-axis represents the potential energy content. If P is at the lower level than the S, the reaction is an exothermic reaction. There is no need to supply energy to such a reaction. Irrespective of whether the reactions are exothermic or spontaneous or endothermic or energy requiring reactions, the S has to go through a much higher energy state or a transition state. The difference in average energy content of S from that of its transition state is called as the activation energy. Now, here you are finding the activation energy within the enzyme, and this is the transition state. So, activation energy for the substrate without an enzyme will be high. When an enzyme is added, it will be low. So enzymes eventually bring down this energy barrier, making the transition of S to P more easy. Now, how do the catalysis happen? First, the substrate binds to the active site of the enzyme, fitting into the active site. Next, the binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate, and new enzyme product complex is formed. The enzyme releases the product of the reaction, and free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Now, let us look at the factors that affect the enzyme activity. One is temperature, second is pH, third is concentration of the substrate. So temperature shows highest activity at the optimum temperature. Activity declines above and below the optimum value. That is why you cannot, higher temperatures also will denature the enzymes. At lower temperatures, enzymes will not be able to work. That is why we had to maintain the correct temperature or the optimal temperature. pH, enzymes will function in a narrow range of pH. Now, even that narrow, if there it is a marked difference in the pH levels, enzymes get denatured. Concentration of the substrate, the velocity of the substrate enzymatic action races with increase in the substrate concentration till it reaches maximum velocity. Further increase of substrate does not increase the reaction rate as there are no free enzyme molecules available to bind to the additional substrate. Now let us look at this graph, you will understand better. So this is with the enzyme action. This is the optimum pH below which it will not be able to function. This is for the temperature and this is the velocity of the reaction and this gives you the Vmax. Now let us look at enzyme classification of okay. Before that, let us finish with enzyme inhibition. When the binding of a chemical shuts off enzyme activity, it is called inhibition, and the chemical is called as an inhibitor. This is because what happens is the inhibitor substance will closely mimic the substrate. As a result of which, it will go and block the active site, and the active site is not able to understand whether it is the substrate that has come to it or whether it is the inhibitor. So it is a mimicked fellow who has come and joined with the active site. So when that happens, then enzyme activity will be suppressed and this is known as competitive inhibition. Example is inhibition of succinic dehydrogenase by malonate. Now let us look at the enzyme classification. This is very, very important because questions will be based on this. Either they will ask you for the description or the definition, or the identify the type from the definition, or give examples. So this table is very, very important. First one is oxidoreductase or dehydrogenase. It catalyzes oxidoreduction between two substrates. Example, alcohol dehydrogenase. It converts primary alcohols to aldehydes. Transferases, that means it transfers, catalyzes a transfer of a group between a pair of substrates. Now, alanine amino transferase shuffles the alpha amino group between the alanine and aspartate. Now, hydrolases, that is breaks, that is hydrolysis of ester, peptide, glycosidic bonds, etc. They are digestive enzymes. Liases, they remove the groups from the substrates by mechanism other than hydrolysis. So they also break, but not with the groups that are not associated with hydrolysis. So decarboxylase removes carbon dioxide from alpha or beta keto acids. Now isomerases catalyzes interconversion of optical, geometrical or position isomer. Example, triose phosphate isomerase. Ligases catalyze linking of two compounds. Enzymes which catalyze joining of carbon, oxygen, carbon, nitrogen, carbon, sulfur, phosphorus, oxygen, etc. Now, let us some things you have to know which are given in your books that is very important with reference to enzymes. One is the cofactors. Enzymes are made up of one or more polypeptide chains. In a number of cases, certain non-protein constituents called cofactors make the enzyme catalytically active. In such cases, protein portion of the enzyme are called the apoenzymes. Catalytic activity is lost when the cofactor is removed from the enzyme. That proves their crucial role in the catalytic activity of the enzyme. Now, three kinds of factors can be identified. One is the prosthetic group, coenzymes, and the metal ions. Now, let us look at the prosthetic group. These are the organic compounds that are distinguished from cofactors by their tight binding capacity to the apoenzyme. For example, in peroxidase and catalases, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is the part of the active center of the enzyme. Coenzymes are also organic compounds, but their association with an apoenzyme is transient. Prosthetic group, it is a tight bonding. If you look at coenzymes, it is transient. It usually occurs during the course of catalysis. Coenzyme serves as cofactors in a number of essential different enzyme catalyzed reactions. Now, essential components of coenzymes are vitamins. Example, if you look at coenzyme nicotinamide adenine dinucleotide NAD and nicotinamide dinucleotide phosphate, the vitamin that is present is the niacin. Now, metal ions A number of enzymes require metal ions for their activity to form coordination bond with side chains at the active site, and at the same time from one or more coordination bonds with the substrate. Example, zinc is the cofactor for proteolytic enzyme carboxypeptidase. Now, this completes the lesson. Now, let us look into the tests. So here we have already clicked on the button. I have already explained how the tests go. Now let us take up first test here. Take a test. Now I understood and I am going to the test here. So amino acids are substituted on the dash carbon. It is alpha carbon. Which of the following is not an aromatic amino acid? Proline. Cytidine is a nucleoside. Dash is not an example of secondary metabolite, uridine, because it's a nucleoside. What percentage of cellular mass is water? 70 to 90%. Adenine, guanine are purines, while cytosine, thymine, and uracil are pyrimidines. Nitrogenous base plus sugar is nucleoside, and its nitrogenous base plus sugar plus phosphate is nucleotide. Protein helices are right-handed. Adult hemoglobin has got 4 subunits. So for this, if you had to look into the test itself, you look into these tables, here you will be able to get the idea. Now if we are talking about human, it has got 2 and 2 chains. That means it has got 4 chains. So that is why I said this test is in congruence with your revision notes and it will help you a lot. Formation of a peptide bond is a dash reaction. So it is a dehydration reaction. In nucleic acid, the bond between the phosphate and hydroxyl group of sugar is a dash bond. We have discussed it as phosphodiester bond. So it is a ester bond. The backbone of DNA is made up of sugar and phosphate. A and T are linked by 2 hydrogen bonds, and G and C have 3 hydrogen bonds. Proteins that catalyze biological reactions are called enzymes. Anabolic pathways consume energy, while catabolic pathways release energy. So I have completed the test. Now all the answers are correct. If in the legend, if this is incorrect, say suppose 3 is incorrect, you will see a red color thing. You just go and click on it. You will be able to see what is the correct answer, and it will also highlight the incorrect answer so that you understand it. And below there will be an explanation to say why it is correct or incorrect. Similarly, if you have not answered, see, one thing is very important. Here you need to understand that in the NEET exams, it is all the entrance exams have negative marking. If you are not sure, don't make a incorrect attempt. If you are not sure, leave the question, doesn't matter, because you are not going to lose any marks. Otherwise, you are going to lose minus 1 mark from your total marks you have scored. Now, if it is yellow, then you can still click understand what is the correct answer along with its explanation. So this is of a great, this would really help you to understand your strengths, your weaknesses, and the way these graphs give you and the things analysis. These analysis will help you to do better and improve your scores in the final exam. Now, with this, we come to an end of today's class. Note that NEET packages have got 38 3-hour full tests with 15 original tests plus 20 past NEET papers, all solved with solutions. You can get additional help by using our Ask feature on our site, and we support chat group using Telegram. You can also book slots for one-to-one teaching with the tutors if you need. And for any enquiries or if you need any questions further that needs answer, mail your query to info@classics.com. Thank you.